Okay, let's go ahead and get started. This is about where we left off with yesterday. Okay.
Yeah, glow. Yeah, glow.
So the main idea behind this is, to learn. But when we're talking about this cell membrane, how many layers does it have? Yeah. Well, you said a couple of them, and I can't remember. So yeah, it has 2 layers to it. Okay. Now, the reason that is so important is because that's what allows to have a cellular boundary to it. What allows materials to come in and come out. We listed 3 things on the board yesterday. I wouldn't expect you to know what they are, but some people might remember what they were. One started with a D, another one an F, and another one an O. Diffusion, facilitated, and then osmosis. Okay? Because the significance of that is blood gases, those are very simple materials that will flow in and out of the cell. That's why it's simple diffusion. Facilitated diffusion, on the other hand, some substances like sugars and other carbohydrates and proteins, they're just too large to enter the cell membrane. So that's why it has to be facilitated. It doesn't take any energy, just like osmosis and simple diffusion doesn't. But then the fourth one, called active transport, that does. So we spent a little bit of time on osmosis and what we call equilibrium. What would happen if you if you only consumed salt water? Will that keep you hydrated?
No, dehydrated.
No, it's going to do just the opposite. You will become dehydrated. In certain cases, yes, it can become fatal. Okay, so moving on then. The structure that, we tried to draw on the board, okay? It's got circular heads with 2 tails on it, okay? One side is polar, the other one nonpolar, okay? So this would just be a guess for yourself. These 3 substances, are they fairly complex? Would it be, what can move in and out of the cell easier? Blood gases or proteins and carbohydrates and cholesterol? Which one is easier? The blood gases or these 3 components? These 3? No. Blood gases are, no, that more so with salt water. No, not with salt water either. Yes, it would have something to do with that because it's a transparent membrane. Yes, so it would have to deal with coffee filters. That's right. Okay? Because when you look at it that way, separates molecules from inside and outside the cell. That's why that plasma membrane is so important. Okay? Let's see here. All right. And it's unfortunate that, and we will get to this later on sometime in this curriculum, Okay? Is it possible to have a group of cells that have absolutely no function at all? It is. Do we possibly know what that is called? It's something that every person, no one wants to get this. And, yeah, it could be a tumor or cancer. Okay? So it's unfortunate that cancer can be part of our daily lives. And it's just cells that have absolutely no function and just take up space. Would we be better off without them? Certainly. So when we talk about a tumor, again, that's something we'd be better off without. I'm trying to think of an example. Let's see here. To be selectively permeable means just that. It's going to allow some materials through that cell membrane and others not. One that might come to mind is something you may take next year or 2 years from now, and that'd be anatomy and physiology. One of the things we talk about there could be a hormone called aldosterone. Okay? And what that hormone does is it allows you to recycle water in the body. But you don't want that aldosterone going into all your cells. You only want that to go into a specific set of cells. So does anyone know where water gets recycled in the human body? We could say there's maybe 2 or 3 sources, but the main one, it's an organ and or gland that starts with a K. Your kidneys will recycle water. So you do not want to have that, again, that aldosterone going into muscle cells or nerve cells, anything to that extent. You only want that to be selective and only go into your renal cells. Renal means kidney. And that's why this cell membrane is so important. And you might also look at like pain medications. Okay? How do they know where to affect pain? How does it know where to go in the human body to alleviate that pain? I just thought about that because I just saw Advil on your tabletop. So how does that Advil know where to go? You think you might have a guess?
There's only one place for it to go.
Well, there is. It's probably where the body is sending a distress signal. Sorry, what was that? Okay, so wherever your body is sending a distress signal, that's calling where that aspirin, ibuprofen, or Advil is going to go. Okay, I want to do this one, this one. I want to get to mitochondria, so just bear with me. Okay. Because then we're going to give you a visual aid that talks about these different types of cell structures and dieting. If we don't get to mitochondria, it's not that big of an issue. I'm trying to keep an eye on the clock at the same time. Well, my point is I don't want to wait till a certain amount of time has elapsed and then show us our visual aid and then run out of time. which I'm trying to read this small print. I'm thinking maybe one more is about all we're going to have time for. Okay, does everyone have the content that wants this? And we're just going to do this last one, then we'll start on the next slide tomorrow. Okay, so ribosomes, as we can see, that's where proteins are manufactured. Proteins are certainly probably the most important, I wouldn't say molecule, but chemical component in our body, because those are enzymes. So if proteins are substances that give us energy, because they're enzymes, where might be a place, where do you think your fastest muscles are? How did you know? You're right. How did you know that? Yeah, that's true. Okay, I would agree. So that's going to take a lot of energy. So you're going to probably have lots and lots of ribosomes in places that require lots of energy. Your eyes, your kidneys, certainly your liver, that's always working hard. Okay? So if there is lots we call rough ER, that means there is lots of ribosomes in that ER structure. If it's smooth ER, then it doesn't have as many of those bumps or ribosomes. Okay, so it's at this time I'll end this presentation as August 22nd, letter A, and we'll pick up with the second section here in just a moment.